Cloning. Scientists using mitosis to their advantage to create new animals from the single somatic cell of an animal that already exists. Continuing to talk about cellular division, today we're going to talk about mitosis and the actual process of dividing from one parent cell into two daughter cells. Now, down here we can see a diagram of the cell cycle, and we can see that only a short portion of it is mitosis. Now, this isn't to scale, so the next diagram will show you an even shorter portion of the cell cycle in mitosis. So mitosis is a small part of the cell cycle. We have G1 phase, S phase, and G2 phase, and these are all known as interphase. So these are going to be between the stages of mitosis. So interphase is divided into G1, which is the growth phase. This is where the cell grows. There's more cytoplasm created. The cell undergoes normal metabolism, and organelles begin to duplicate. So we have more mitochondria, more ribosomes, more vacuoles, and more lysomes during this phase. During the S phase, known as synthesis, DNA replication occurs. So we go from having one of each chromosome to two of each chromosome. So the chromosomes duplicate and the cell continues to grow in size. During the G2 phase, growth also occurs, and this is in preparation for mitosis. So the necessary enzymes are going to be created, as well as increasing the amount of cytoplasm so that we have two daughter cells with the requisite amount of cytoplasm, enzymes, and organelles for successful development. So mitosis is both a nuclear and cytoplasmic division of the cell into two identical daughter cells. So nuclear meaning like nucleus and genetic material. It's a function of growth and repair in multicellular organisms. So this allows our hair to grow longer, our bones to grow, our muscles to get stronger, our fingernails to continue to grow, cuts to heal, blood vessels to heal. So all those different times where our body needs to grow or fix itself, it's due to mitosis. And this happens in both asexual, it's a function of growth and repair in multicellular organisms. And this would be the type of asexual reproduction in unicellular eukaryotic organisms. So if an organism is only one cell, mitosis is the process by which it makes more of itself. So in animal cells, mitosis is divided into four phases. And interphase is very important. So we need interphase for mitosis to proceed, but interphase is not one of the phases of mitosis. So the first phase of mitosis is known as prophase. So this is when the chromatin condenses to form chromosomes consisting of two sister chromatids connected by a centromere. So we'll see them in that X pattern. The nuclear membrane will begin to dissolve. So this would be the nuclear membrane around the nucleus of the cell, like the organelle of the nucleus. And centrioles, which are organelles that provide attachment to spindle fibers, are going to move to the opposite ends of the cell. So these are where spindle fibers will come down, and they're going to attach to one of each sister chromatid, and they're going to pull them to separate ends in later phases. But we'll see those centrioles form with some spindle fibers that are attached to them. So this is what a diagram of a cell in prophase. We can see that there still is remnants of a nuclear envelope. We see the chromosomes formed into X-shaped patterns, each with a little black centromere. Our centrioles have moved to the poles, and spindle fibers are beginning to reach down to grab onto the chromosomes. So not every prophase image will look the same, but some things to look out for are those centrioles moving towards or near the poles. Sometimes you'll see them farther away from the poles, but there'll still be two of them, chromosomes formed into X's, and the remnants of a nuclear envelope. The second stage of mitosis is metaphase, and the key to these images are where the chromosomes line up at the middle of the cell along the equatorial plate. So if you think about the equator on a globe, it's around the center. Metaphase starts with M, middle starts with M, and then the spindle fibers will attach to each centromere of the chromosome. So we'll see that on the next slide. So here we can see that the chromosomes have lined up along the middle of the cell, also known as the equatorial plate, and a spindle fiber has attached to the centromere of each chromosome. The next phase of mitosis is known as anaphase. Now, the centromere, now holding the two sister chromatomes together, has divided or split, and each sister chromatid, which now is called an individual chromosome, becomes pulled to the opposite ends of the cell. And now there's two identical sets of chromosomes, one at one of the poles, one at the other of the poles. So when you're looking at images of anaphase, as a diagram, an important note is that all of the sister chromatids have now been separated. One is being pulled by a spindle fiber to each end of the cell. So those sister chromatids have split. Now it's each of them is a single chromosome. They're being pulled to either end of the cell by the spindle fibers. The last phase of mitosis is telophase. So this is when the chromosomes begin to decondense, become chromatin again. The nuclear membrane will form around each mass of chromatin, so you'll probably see two nuclear membranes forming in an image. And a cleavage furrow begins to form as the membrane pinches off. So the cell starting to pinch itself into two. Now imagine you're kind of grabbing a marshmallow and trying to rip it into two pieces, and how you pinch in the middle and it starts to get skinnier. And this process is known as cytokinesis. Now in the next slide, we don't actually see any cytokinesis occurring. But we can see that the chromosomes are now decondensing into chromatin, and we have two nuclear envelopes forming in the cell, and the spindle fibers have detached from the individual chromosomes. So here is our flow chart of mitosis. Now, a nice little acronym to remember that is PMAT. So prophase, metaphase, anaphase, telophase, and then the last picture here is cytokinesis. So sometimes you'll see pictures similar to the one that's identified as cytokinesis, and that will be the picture that you would have to identify as telophase. So the telophase could be one of the bottom two pictures in any given diagram. So again, prophase, we see those chromosomes are in an X pattern, and the nuclear envelope has started to decondense. Metaphase, the chromosomes that line up at the equatorial plate in the middle. Anaphase, they have split into their individual sister chromatids, and they're being pulled to each pole. And in telophase, we have two nuclear envelopes forming, and the process of cytokinesis, or the physical cell division, has started to take place. So overall, the process of mitosis produces two daughter cells that are genetically identical to the parent cell, forming genetic continuity within an organism. So if one of my skin cells divides, it creates two skin cells that are identical to the parent cell. In plants, mitosis is different because the plant cells lack centrioles, but spindle fibers still form and serve a similar purpose. And then because of a cell wall, cytokinesis doesn't occur, but a new cell plate ends up forming between those two masses of chromatin. And the amount of time a cell spends in mitosis or interphase depends on the type of cell. So some cells will divide much more quickly than others, but cells can only divide a finite number of times, which is why cells will age and will get things like wrinkles and saggy skin, or organs will start to deteriorate because those cells have replicated their maximum number of times. And different cells have different maximum numbers of replications available to them. So cloning is a really cool process by which identical offspring are formed from a single somatic or body cell. So this doesn't use gametes or sperm or egg cells. And the offspring are identical to the parent cell unless a random genetic mutation happens to take place. So technically, cloning is asexual reproduction. Several organisms use this as a way to reproduce. So hydra that make reproduce through budding, where they'll grow kind of like a mini one of themselves that'll eventually break off. Or strawberry plants, which produce runners. And then a new strawberry plant will grow, and it's going to be identical to the parent plant. Since roughly the 1950s, scientists have been experimenting with cloning complex organisms, and the first successful clone was Dolly the sheep in 1996. And they cloned Dolly from the udder of her cell donor. The cloning depends on the ability of whether the parent cell or the donor cell is totipotent. So does this cell have the ability to divide and differentiate into an entire organism? So to make complex organisms cells totipotent, they end up providing them with a lot of electric shocks and currents so that it becomes totipotent. Because typically, like an udder cell for a sheep would only divide into other udder cells. There are some organisms like starfish are able to grow new arms, and lizards that can generate new tails have totipotent cells. But human cells lose this ability after roughly the third mitotic division. So after the morula forms 8 cells, it's already beginning to specialize. And if you took any one of those 8 cells out, it may or may not have the ability to form an entire organism. And then even if you look at past day 5, but like 6 to 12 in that process of gastrulation, cells have really already started to specialize. So when we look at cell death and aging, some cells have the ability to divide faster than others. So red blood cells live only about 120 days, and nerve cells in the brain do not divide at all. And all cells have a biological clock that regulates the number of divisions available to them. And once it completes those number of divisions, it isn't able to successfully reproduce again. It's not able to produce more daughter cells. And so skin cells, blood cells, cells of the digestive tract reproduce more often than our specialized muscle cells, nerve cells, and secretory cells. Only spermatocytes and cancer cells can escape cell aging. So sperm can be produced roughly throughout a man's life from when puberty starts. And cancer cells are cancer because of their inability to stop dividing. So cancer is a rapid, uncontrolled cell division. And the abnormal cell division is due to a mutation in the cancer-causing genes, or like an oncogene, the cancer-causing gene. So they differ from normal cells because they divide way too fast to specialize. So they're non-functional cells. And they can divide when separated from the other cells. So they can, if you had a tumor somewhere in your body, cells can dislodge from this and travel throughout your body and become cancer somewhere else. And this is known as metastasis. So if your cancer has metastasized, it may have started in your breasts, but then it has traveled through your body, and now you might have lung cancer or liver cancer. They don't change shape as they mature, and the nucleus tends to be enlarged in a cancer cell. The liver is also really interesting because it performs body functions like maintaining constant levels of blood sugar and detoxifying the blood. But liver tissue can continue to grow even after transplant. So if it's a whole liver has been cut into 2 and maybe be transplanted into 2 people, it can continue to grow and divide into a normal liver size. So that's something that's really special about liver function in humans. Also, if you have some liver scar tissue, your liver is much more able to repair itself than other body organelles. So that's our lesson on mitosis. Enjoy.